Welcome back to the deep dive. If you're tuning in, you're probably looking for that edge, that shortcut to being really well informed. And today we've got something pretty specific lined up. This one's for the students in Dr. Rami's BioSci 221 microbiology course over at College of the Canyons.
That's right.
Our mission today, to get you prepped and ready, focusing on the key stuff you'll need for lecture quiz, #1A.
Exactly. Think of this as your high-yield study partner.
Perfect. So let's unpack this.
Yeah, we're going to basically boil down the essentials from Chapter 1, the Introduction for Health Professions, and Chapter 2, which is all about bacteria, their structure, function, and, critically, their clinical relevance.
And this isn't just cramming for a quiz, right?
Absolutely not. This is foundational. It's the bedrock knowledge every registered nurse needs. We're aiming for real understanding here because you will use this stuff daily in your practice.
Okay, so let's start right there. The big question, why? Why does understanding this microscopic world matter so much for someone going into nursing? It could seem a bit abstract at first.
It really could, but it's about as far from abstract as you can get in practice. As a nurse, you're literally the first line of defense against infections.
Okay.
Think about your daily tasks, drawing blood, inserting catheters, giving antibiotics, even just educating patients. Microbiology is woven into every single one.
Right.
Preventing hospital-acquired infections, HAIs, that comes down to understanding the body's own microbes, the microbiome, and knowing how to maintain a sterile field.
Makes sense.
And recognizing infection signs. Is that cough viral? Or is it bacterial pneumonia? Knowing the difference helps you advocate for the right treatment or, crucially, against unnecessary antibiotics because, they're useless against viruses.
A huge issue, antibiotic resistance.
Exactly. And interpreting lab reports, seeing Staphylococcus aureus on a wound culture, you need to know instantly why that matters. Or explaining to a patient why finishing their entire antibiotic course is non-negotiable.
Or why vaccines work.
Or why vaccines work, precisely. It's not just another science class. It's your practical toolkit for keeping patients safe.
That really grounds it. Okay, so with that context, let's dive into the microbial world itself. It's huge, diverse, mostly invisible. What kind of players are we talking about?
It's a whole cast of characters, mostly microscopic. We can sort of group them. First, you've got the acellular microbes, not really cells in the traditional sense.
Viruses.
Exactly. Viruses are basically tiny bundles of genetic stuff, DNA or RNA, inside a protein coat. They're tiny, they can be under 200 nanometers, and they have to infect a host cell to replicate. They're obligate intracellular parasites. Flu, HIV, SARS-CoV-2, classic examples. Even simpler and maybe scarier are prions. These are just infectious proteins that have misfolded. No genetic material at all, under 10 nanometers. But they cause awful neurodegenerative diseases like Creutzfeldt-Jakob disease, and they're incredibly hard to destroy.
Just protein. Wow.
Then we get to the actual cells, the prokaryotes. These guys lack a true nucleus.
Bacteria fall in here, right?
Correct. Bacteria are the stars in many ways. Single-celled, usually around 1 micrometer. They have cell walls made of peptidyl glycan, and they multiply by binary fission, just splitting in two.
Yeah.
Think Staph aureus, E. coli, Mycobacterium tuberculosis.
The ones we hear about a lot.
Yeah. And their cousins, the archaea. Also prokaryotic, similar in size, but their cell walls are different. No peptidoglycan. glycan. Many are extremophiles living in harsh places. Key point for you. No known human pathogens among archaea.
Good to know.
Finally, the eukaryotes. These do have a nucleus and organelles, like our cells. This includes protozoa, single-celled, often motile guys, like Plasmodium that causes malaria.
Bigger, too.
Generally, 10 to 100 micrometers. Then fungi, which includes single-celled yeasts, like Candida that causes thrush, and multicellular molds, like Aspergillus. They have chitin in their cell walls.
And worms, are they microbes?
Helminths. The parasitic worms themselves can be huge, meters long even. But their eggs and larval stages are often microscopic, less than 200 micrometers. And that's how they're transmitted and diagnosed. So yes, relevant to microbiology in that sense.
It's a huge range. And you mentioned earlier this idea of the microbiome, that these aren't just invaders. They're living on us and in us.
Absolute critical concept. Our bodies are teeming with microbes. Skin, gut, mouth, everywhere. This community is the human microbiome or microbiota. And mostly they're beneficial or at least neutral.
Oh, so?
They help digest food, they synthesize vitamins, they actually train our immune system, and there's growing evidence they even influence mood. It's a complex ecosystem.
But it can go wrong.
Definitely. The balance is key. Take antibiotics, for example. Broad-spectrum ones can wipe out a lot of the good gut bacteria along with the bad. This disruption creates an opportunity for potentially harmful microbes that were being kept in check to overgrow.
Like CDiff.
Exactly, the prime example, Clostridioides difficile. It can cause really severe diarrhea, colitis, especially after antibiotic treatment. So, as a nurse, if your patient on antibiotics develops diarrhea, C. diff should be on your radar. You'll monitor for it, maybe advocate for probiotics, or understand why something drastic like a fecal transplant might be needed. It connects directly back to patient observation and care.
That's a perfect clinical link. It really shows how this isn't just theoretical. Okay, shifting gears slightly. How did we even figure all this stuff out? Let's touch on some history. These discoveries didn't happen overnight.
Not at all. It's a fascinating story built over centuries. You go back to Girolamo Fracastoro in, believe it or not, 1546. He proposed these invisible contagion particles, a very early hint of germ theory.
Wow, way before microscopes could really see them.
Right. Then Anthony van Leeuwenhoek, around 1675. He was the Dutch draper, not a scientist by trade, but he ground amazing lenses and built powerful single-lens microscopes. He was the first to actually see and describe bacteria and protozoa, his animalcules. He opened the door. Jump to the mid-1800s, things really accelerate. Ignaz Semmelweis, 1847. A physician who noticed women dying from childbed fever at alarming rates in one clinic compared to another. He hypothesized cadaverous particles were being transferred by doctors from autopsies.
And he pushed for hand washing.
Yes, with chlorinated lime solution.
Yeah.
And the death rates plummeted. It seems obvious now, but he faced huge resistance. It's the foundation of modern hand hygiene.
Incredible. And tragic he wasn't listened to initially.
It was. Then you have John Snow in London, 1854. During a cholera outbreak, he didn't just treat patients, he mapped the cases. He traced the source to a specific public water pump on Broad Street. Removing the pump handle stopped the outbreak. That's basically the birth of epidemiology. Public health intervention based on evidence.
So clever, using data.
And then the giant, Louis Pasteur. Mid to late 19th century, he definitively disproved spontaneous generation, the idea that life just arises from nothing. He showed microbes cause fermentation, developed pasteurization to kill microbes in wine and milk, and created vaccines for rabies and anthrax. He solidified the germ theory of disease.
A true game changer.
Absolutely. And building on that, Joseph Lister, around 1865, he applied germ theory to surgery. He started using carbolic acid phenol to sterilize instruments and clean wounds. This introduced antiseptic surgery, making operations far safer.
Reduced infections dramatically, I bet.
Hugely. Then Robert Koch, starting in the 1870s, he gave us the tools to prove which microbe causes which disease. He developed techniques for growing bacteria in pure culture on solid media. And he formulated Koch's postulates, a systematic set of criteria. Find the microbe in diseased animals, isolate it, infect a healthy animal, cause the same disease, and re-isolate the same microbe.
The scientific method for infectious disease.
Precisely. He used it to identify the causative agents of anthrax, tuberculosis, and cholera. It established diagnostic microbiology. While we have molecular updates now, the core logic remains.
And then finally, fighting back with drugs.
Yes. Paul Ehrlich searched for a magic bullet and found salvarsan for syphilis in 1909, early chemotherapy. But the revolution was Alexander Fleming in 1928, noticing that penicillium mold killed bacteria on his Petri dish.
The famous accident?
The famous fortunate accident. But it took Howard Florey and Ernst Chain in the 1940s to figure out how to purify penicillin and scale up production just in time for World War II. It ushered in the antibiotic era, followed by Waxman and others finding more antibiotics like streptomycin.
It's just amazing how each discovery built on the last. And all of this directly informs how nurses practice today: infection control, surgery, treatments.
Exactly. It's not just history, it's the why behind what you do every day.
Okay, so from history to the lab bench and the bedside, let's talk practical tools and safety. How do we actually see and work with these microbes safely?
Right, the tools. Microscopy is obviously central. The standard is the bright field microscope. Uses visible light, passes through objective lenses and ocular lenses to magnify. For the highest power, 100x objective, you need immersion oil to improve resolution.
The one everyone uses in lab?
Pretty much. But there are others. Dark field microscopy is clever. It blocks central light, so the specimen is lit only from the sides against a dark background. Great for seeing thin, unstained microbes like Spirochaetes.
Like the one that causes syphilis.
Exactly, Treponema pallidum. In phase contrast microscopy, enhances the contrast between parts of the cell with different refractive indices. It's fantastic for looking at living, unstained cells, seeing internal structures or motility.
And seeing them often involves staining, right? You mentioned that earlier.
Yes, staining adds contrast and helps differentiate. A simple stain uses just one dye basic info on shape and arrangement. But the powerhouse is the gram stain.
The purple and pink one.
That's the one. It separates bacteria based on their cell wall structure into gram-positive, purple, thick peptidoglycan, and gram-negative, pink, thin peptidoglycan plus an outer membrane. This is often the first piece of identifying information from the lab.
And it helps guide treatment fast.
Immensely. It guides empiric therapy, the initial antibiotic choice before the specific species is known. Knowing if it's gram-positive cachet versus gram-negative rods points you in very different directions.
What other stains are important?
The acid-fast stain is crucial for identifying mycobacterium species, like M. tuberculosis, with their waxy mycolic acid walls. Then there are special stains to highlight specific structures if needed: capsule stains, endospore stains, flagella stains. They help identify virulence factors.
Okay, we can see them. Now, how do we handle them safely, both in the lab and clinically? You hear terms like sterile and disinfect.
Terminology here is key for nurses. Aseptic technique is the overarching principle, all the practices you use to prevent unwanted microbial contamination. Hand hygiene, using sterile gloves, prepping skin before injection. Foundational basics. Sterilization means killing everything, all microbes including viruses and those tough endospores. Think autoclaving instruments. Disinfection eliminates most pathogens from inanimate objects like wiping down a counter, but it might not kill spores.
Okay.
Antisepsis is similar to disinfection, but for living tissue, like using an alcohol wipe or chlorhexidine on skin before drawing blood. Degerming is mostly mechanical removal, like scrubbing during hand washing. And sanitization just lowers microbial counts to what's considered safe for public health, like washing dishes.
Clear distinctions. And what about those BSL levels you hear about labs using?
The biological safety levels.
Yeah.
They classify labs and procedures based on the risk of the microbes being handled. 4 levels. BSL-1 is for low-risk microbes, not known to cause disease in healthy adults. Basic teaching labs.
Like non-pathogenic E. coli.
Exactly. BSL-2 is for moderate-risk agents, common human pathogens that we have treatments for, like Staph aureus or Salmonella. Requires protective gear like lab coats, gloves, eye protection. It usually works in biological safety cabinets. Most clinical labs operate at BSL-2.
Makes sense.
BSL-3 is for serious or potentially lethal diseases, especially those transmitted by aerosols, like Mycobacterium tuberculosis. Requires restricted access, negative airflow rooms, respirators, more specialized labs.
Okay.
And BSL-4 is the maximum containment level for dangerous exotic agents with high fatality rates and often no treatments, like Ebola or Marburg virus. Think full body, air-supplied suits, isolated buildings.
Highly specialized, so nurses mostly interact with BSL-2 level concerns.
Primarily, yes, in terms of the pathogens they encounter directly. But understanding the BSL system helps you appreciate the infection control measures required for certain diseases, like TB needing airborne precautions, which stems from its BSL-3 nature.
Got it. Okay, so we have all these different microbes. How do scientists keep them organized? The naming seems complicated.
It's systematic, thankfully. Taxonomy is the science of classification. The big picture now is the 3-domain system bacteria, archaea, and eukarya, proposed by Woese and Fox, based on ribosomal RNA sequences. Very fundamental.
And the specific names, like Staphylococcus aureus.
That's binomial nomenclature, developed by Linnaeus. Every organism gets a unique 2-part name. The first part is the genus, always capitalized. The second is the specific epithet, always lowercase. Both are italicized or underlined.
So Staphylococcus is the genus, aureus is the specific epithet.
Precisely. And after you mention it once, you can abbreviate the genus, like S. aureus. Why does this matter clinically? Accuracy. Confusing Staphylococcus epidermidis, a common, usually harmless skin bacterium, with S. aureus, a major pathogen, could lead to incorrect treatment. Precision in naming is crucial for communication.
Makes perfect sense. All right, let's really zero in on bacteria now. They seem to be the focus for a lot of infectious diseases relevant to nursing. What makes them tick structurally?
Okay, bacteria basics. They're prokaryotes, remember. No nucleus. Their genetic material is usually a single circular chromosome located in a region called the nucleoid. They also have ribosomes, slightly different from ours, called 70S ribosomes, which are essential for making proteins.
And those plasmids you mentioned.
Right. Many bacteria also have plasmids. These are small, circular, extra pieces of DNA separate from the chromosome. They replicate independently and often carry genes that give the bacteria an advantage, like antibiotic resistance or factors that help cause disease, virulence factors.
What about the outer layers? You mentioned the cell wall is key.
Absolutely key. The bacterial cell wall provides structural support, maintains shape, and critically protects against osmotic lysis. Bacteria often live in environments more dilute than their cytoplasm, so water constantly tries to flow in. Without the rigid wall, they'd swell and burst.
Like our red blood cells would in pure water.
Exactly. Our cells lack walls. Is dangerous. It can apollute their red blood cells. Conversely, a hypertonic solution makes them shrivel, crenate. We use isotonic solutions, like 0.9% saline, because they match the solute concentration of our blood, preventing water from rushing in or out of cells. This clinical practice is directly related to understanding bacterial osmotic protection.
That's a fantastic connection. And the wall itself differs, leading to the Gram stain result.
Correct. Gram-positive bacteria have a thick cell wall, mostly peptidoglycan, like 30 and 100 millimeters thick.
Yep.
It also contains teichoic acids and lipoteichoic acids, which anchor the wall and can trigger inflammation. This thick wall traps the crystal violet iodine complex during the Gram stain, so they look purple.
Okay. And Gram-negative?
Gram-negative bacteria have a much thinner layer of peptidoglycan, 4 nanometer. But, and this is crucial, they have an outer membrane outside the peptidoglycan layer. This outer membrane is unique. It contains lipopolysaccharide, LPS, which is a major virulence factor we'll come back to. It also has porins, protein channels, and creates a periplasmic space between the inner and outer membranes. This structure doesn't hold the crystal violet, it gets washed out, and they pick up the pink counterstain, safranin.
That outer membrane sounds important.
Very. It acts as a barrier, blocking entries of antibiotics and disinfectants. And don't forget acid-fast bacteria like Mycobacterium. They have peptidoglycan, but it's linked to other complex molecules and overlaid with a thick, waxy coat of mycolic acids. This makes them resistant to drying, disinfectants, and requires that special acid-fast stain.
Beyond the wall, what about their shapes and any, accessories they might have?
Shape or morphology is a primary characteristic. Coccus means spherical. They can arrange in pairs, diplococci, chains, trepicoci, or grape-like clusters, staphylococi. Bacillus means rod-shaped. These can be short, coccobacilli, long, curved.
Vibrio for curved.
Yep, vibrio is a curved or comma-shaped rod. Then you have spirals. Spirillum is a rigid spiral, usually with external flagella. Spirochetes are flexible, corkscrew-shaped bacteria with internal flagella, endoflagella, that let them move in a unique way. Think Treponema pallidum again.
And these arrangements, like chains or clusters, tell you something.
They do. They result from how the cells divide and whether they stick together. Combined with Gram stain and shape, these arrangements give rapid clues. Gram-positive cocci in clusters. Think Staphylococcus. Gram-positive cocci in chains. Think Streptococcus. It helps narrow down the possibilities quickly for that empiric therapy.
Okay, what about external structures, things sticking out?
Many bacteria have a glycocalyx, a sticky layer outside the cell wall, usually made of polysaccharides. If it's well organized and firmly attached, it's called a capsule. Capsules are major virulence factors. They help bacteria resist phagocytosis by immune cells, stick to surfaces, and prevent drying. Streptococcus pneumonia, the common cause of pneumonia, often has a capsule.
Slime layer is looser.
Exactly. A slime layer is looser, less organized. Both capsules and slime layers can help form biofilms, communities of bacteria encased in this matrix, which makes them very hard to treat with antibiotics.
Like on catheters or implants.
Precisely. Then there are protein appendages. Fimbriae are short, bristle-like fibers used for attachment to surfaces or host cells, essential for colonization. Pili are similar, often longer, and a specialized type, the sex pilus or F. pilus, is used to transfer DNA between bacteria during conjugation.
Sharing those resistance genes.
And finally, flagella. These are long, whip-like tails used for motility. Bacteria can have one, monotricious, one at each end, have fitritrous tufts at one end, lofitricious, or all over paratricious. Motility itself can be a virulence factor, helping bacteria spread, like Vibrio cholerae moving through mucus.
It's incredible how specialized these tiny things are for survival and sometimes causing trouble. Let's talk more about that survival strategies and clinical impact. Endospores sound particularly tough.
They are nature's ultimate survival pods. Endospores are dormant structures formed inside the bacterial cell by certain gram-positive rods, mainly Bacillus and Clostridium genera. They're not for reproduction, but for survival.
What do they resist?
Almost everything. Extreme heat, drying, UV radiation, freezing, lack of nutrients, many chemical disinfectants. They can survive for decades, even centuries. waiting for conditions to become favorable again to germinate back into a vegetative active cell.
Which bacteria make them that are clinically important?
Several big ones. Bacillus anthracis, anthrax, Clostridium tetanus, Clostridium botulinum, botulism, Clostridium perfringens, gas gangrene, and the one we mentioned, Clostridioides difficile colitis.
So for nurses, this means?
It means standard disinfection might not be enough if spores are suspected. You need sporicidal agents, often bleach-based. And crucially, alcohol-based hand sanitizers do not kill C. diff spores. You need thorough hand washing with soap and water to physically remove them. Huge infection control point.
Got it. And you mentioned LPS and gram-negatives earlier. What's the big deal with that clinically?
Lipopolysaccharide, LPS. Specifically the lipid A portion, which anchors LPS in the outer membrane. When gram-negative bacteria die, often when killed by antibiotics in big part, lipid A is released. It acts as a potent endotoxin.
Endo, meaning within the bacteria, released upon death.
Correct. Unlike exotoxins, which are actively secreted, this lipid A triggers a massive inflammatory cascade in the host. Fever, chills, aches, a drop in blood pressure due to vasodilation. It can lead to septic shock, which has a high mortality rate.
So recognizing a gram-negative infection in someone becoming septic is critical.
Absolutely. It alerts you to the potential for this endotoxin-mediated shock. It reinforces the need for rapid diagnosis, aggressive fluid resuscitation to counter the vasodilation, and appropriate antibiotics to clear the infection causing the endotoxin release in the first place.
Okay, and that brings us back to antibiotic resistance. Plasmids and sharing genes sound like a major reason this is such a problem.
It's a huge driver. Remember plasmids, those extra DNA circles? They often carry R genes, resistance genes, and bacteria are promiscuous with their DNA. They share these plasmids readily through horizontal gene transfer, HTT.
The three ways.
Yep. Conjugation is direct transfer, like bacterial sex using that pilus. Transformation is picking up naked DNA fragments released from dead bacteria in the environment. Transduction uses bacteriophages, viruses that infect bacteria as fairies to accidentally transfer bacterial DNA, including resistance genes, from one bacterium to another.
So resistance can spread incredibly fast.
Frighteningly fast. This explains the rapid emergence of multi-drug-resistant organisms, MDROs, like MRSA, methicillin-resistant Staphylococcus aureus, or carbapenem-resistant Enterobacteriaceae, like Klebsiella. It underscores why we must use antibiotics judiciously, antibiotic stewardship, and maintain strict infection control to prevent the spread of these resistant strains.
And just quickly, how do antibiotics actually work? Why don't they harm our own cells?
That's the principle of selective toxicity. Antibiotics target structures or pathways that are unique to bacteria or significantly different from our eukaryotic cells.
Like the cell wall.
Exactly. Peptidoglycan synthesis is a prime target. Beta-lactam antibiotics, penicillin, cephalosporins, and vancomycin disrupt this process. Since we don't have peptidoglycan, these drugs are generally safe for us. They tend to be more effective against gram-positives with their thick peptidoglycan layer.
What else?
Bacterial protein synthesis. Bacteria have 70S ribosomes, while ours are 80S. Drugs like aminoglycosides, tetracyclines, and macrolides bind to the 70S ribosome and inhibit protein production. Others target nucleic acid synthesis. Fluoroquinolones block DNA gyrase. Rifampins block RNA polymerase. Some block essential metabolic pathways like folate synthesis, sulfonamides, trimethoprim, which bacteria make but we get from our diet, or mycolic acid synthesis, targeted by isoniazid for TB. A few even disrupt the cell membrane, like polymyxins against gram-negative outer membranes.
Knowing the target helps choose the right drug.
Yes, and explains limitations. For example, vancomycin is large and can't get through the outer membrane of gram-negatives, limiting its use primarily to gram-positive infections.
This is tying so much together. Okay, final stretch. Let's bring it right to the bedside. For a future RN, what are some of the absolute headliner bacteria they need to know cold? The frequent flyers, so to speak.
Okay, rapid-fire key pathogens. Knowing the basics here is high yield for your quiz and your career. Staphylococcus aureus, including MRSA. Gram-positive Kaji, clusters. Normal on skin O's for many, but causes everything from boils to pneumonia, bone infections, sepsis. MRSA is the resistant variant, a huge HAI concern. Watch out for transmission via surfaces, hands, devices.
Got it. Clusters staph, Streptococcus pyogenes, group A strep. Gram-positive Kachi chains, think strep throat, scarlet fever, skin stuff like impetigo, but also serious things like necrotizing fasciitis, flesh-eating bacteria. Spreads via droplets contact, chains strep. Streptococcus pneumonia, pneumococcus. Gram-positive, often lancet-shaped pairs, Diplococci usually has a capsule. Major cause of community-acquired pneumonia, also meningitis, ear infections. Vaccine preventable, thankfully. Okay. Clostridioides difficile, CDiff, gram-positive spore-forming rod, anaerobic, causes antibiotic-associated diarrhea colitis. Remember, spores resist alcohol gels. Wash hands with soap and water, bleach for environmental cleaning. Spores, hand washing. Escherichia coli, E. coli. Gram-negative rod, mostly harmless gut bug, but pathogenic strains are the #1 cause of UTIs, also cause various diarrheas, like the dangerous O157H7 strain, and can cause sepsis. Fecal-oral transmission common, gram-negative rod very common. Klebsiella pneumonia, gram-negative rod, often has a big capsule, causes pneumonia, especially in hospitals, UTIs, bloodstream infections, worrying trend of carbapenem-resistant strains, CRKP. Strict infection control needed, another resistant gram-negative, Pseudomonas aeruginosa, gram-negative rod, often motile, loves moist environments, hospitals, opportunist, infects burns, lungs, especially in CF patients, urinary tracts via catheters, forms biofilms, naturally resistant to many drugs, the green pigment one sometimes.
Sometimes, yes. Distinctive smell, too. Mycobacterium tuberculosis, acid-fast rod, causes TB, spread by airborne droplets, needs special diagnostics, acid-fast stain, prolonged multi-drug treatment, and airborne isolation precautions for patients with active pulmonary TB.
Acid-fast equals TB. Special precautions.
And there are others, of course: salmonella, food poisoning, Neisseria gonorrhea, Campylobacter, gastroenteritis, Vibrio cholerae, cholera, Listeria, foodborne illness, dangerous in pregnancy. Knowing the diagram reaction, shape, key disease, and maybe one critical nursing point, like spores, resistance, transmission, for each is a great foundation.
That's an excellent wrap-up of the key players. We've really covered a lot today: the types of microbes, the history, the tools, bacterial structures, survival tactics, and the pathogens nurses need to know.
We have. And the core message really is that this isn't just abstract science you memorize for a test. It directly informs almost everything you'll do to care for patients safely and effectively. From washing your hands, to understanding lab results, to administering antibiotics correctly, to patient education, it all connects back.
The microbiome as friend and foe, the legacy of Semmelweis and Pasteur in our daily routines, the challenge of resistance.
Exactly. It all raises a really important question, doesn't it?
It does. So as you prepare for this quiz, but more importantly, as you look towards your career as an RN, think about this. How will you take this knowledge, this understanding of the microbial world, and use it? How will you use it to protect your patients, to be smarter in your practice, to be a stronger advocate against infection, and ultimately to make a real difference in the fight against these invisible but ever-present threats? Think about the impact you can have.